Hey everyone, this is Snigdha. Today I'll be presenting my research question, which is there an association between high dietary glycemic index, glycemic load, and the risk of developing gynecological cancer in women? So, a short introduction to who I am. Currently tuning in from Bangalore, India, and I have a huge passion for taking cancer biology and gynecology, and I need to pursue a career in medicine. Therefore, this project has given me the opportunity to explore all these various interests that I have and sort of combine it into one paper. And my mentor, Dr. Kim Price, has been incredibly helpful in guiding me through this process. So, why exactly did I choose this topic? Well, the excessive consumption of refined and simple carbohydrates has become a trend since the beginning of the 21st century, and its association with diseases like cardiovascular disease and obesity is well known. However, its association with cancer is something that is less explored, specifically gynecological cancer. And hence, through this project, I aim to explore how such a diet, which is very relevant to my culture as well, has an effect on women's health. A little bit of context first. What exactly happens in a high carbohydrate diet? Well, this is defined as a diet where greater than 60% of one's total dietary energy comes from carbohydrates. When this happens, we eat a meal and there happens to be a lot of carbohydrates in it. We end up having high levels of glucose in our blood, which results in higher levels of insulin in our blood, a hormone that is responsible for regulating the amount of glucose in our blood. However, when we do this chronically over a long period of time, this ends up leading to our body become desensitized to insulin, which leads to insulin resistance. And so, while the body may not be able to effectively regulate glucose levels, there is an excessive amount of insulin in your blood, which is called hyperinsulinemia. And it's dangerous because insulin acts as a mitogen, which is essentially a substance that increases the rate of a cell dividing, that is mitosis, which is the starting point of all cancer. And how do carbohydrates link with cancer? Well, I said that insulin is a mitogen. So how this exactly happens is that it binds to insulin receptor and triggers multiple pathways. Two of them, which are listed here, involve a cascade of proteins, which ultimately result in an increase in protein proliferation and decreased apoptosis, that is, regulated self-induced cell death. And so at the same time, there's also insulin-mediated metabolic effects. However, when there is insulin resistance, the insulin-mediated metabolic effects are reduced while cell growth is still at a high point and still increasing, leading to creation of tumors. And so this is linked with gynecological cancers, which is essentially just cancer starting in the female reproductive organs located within the pelvis. And there are many types of GCs, which are cervical, ovarian, and uterine, and they share similar risk factors, preventions, and treatments. And how this links back to insulinemia is the fact that high levels of insulin can lead to free unbound sex hormones like estrogen. And you may have heard that estrogen is especially dangerous and is seen in breast cancer because it acts as a human carcinogen, which means it increases the risk of a cell becoming a cancer cell. So the hypothesis that I came up with based on this background information is the risk of developing gynecological cancer is positively associated with high glycemic index and glycemic load. To sort of break this down, the exposures are the high glycemic index and load. Glycemic index is essentially just a scale between 1 and 100, which measures the level of glucose in your blood after you eat a meal. and 100 being when you eat just plain sugar, glucose. Glycemic load is a more accurate measure as it also takes into account the quantitative amount of glucose or carbohydrates in a particular meal. And the outcome that I'll be investigating is the incidence of gynecological cancer. So the methods I used and the tools I used were based upon conducting a meta-analysis. And what a meta-analysis essentially is a quantitative synthesis of studies that share a similar research question. Why meta-analysis is specifically helpful is because it increases the sample size and statistical power while also bridging conflicting studies and conclusions. In the process, it produces new data and conclusions based on powerful statistical techniques, and is usually more reliable than just taking the conclusions of an individual trial or epidemiological study. The process of a meta-analysis is pretty robust and has included a literature search, which is searching multiple databases such as Google Scholar, PubMed, Cochrane Library, and sort of sifting through all the research papers based on the keywords that you input. And in the process, you end up after further screening the abstracts and screening the papers, I ended up with 14 studies that I used in the final data set. And so the data collection from these individual papers, I collected the effect size, which are values that quantify the effect that glycemic index has on GC risk. Confounding variables, which are essentially other risk factors that also contribute to GC risk, and confidence intervals, which is just a margin of error that tells us whether there is truly a positive or negative effect of glycemic index on GC risk. So to conduct the actual meta-analysis, I used a software called JASP, a user-friendly software that uses multiple statistical tests, and I used the random effects model, which is essentially including of 3 statistical tests that I will be discussing in the results. So, was the hypothesis falsified? We can see this through 2 diagrammatic representations of the results, which are called forest plots. On the left, we see a glycemic index, effective glycemic index on GC risk, and we can see that the overall effect from the random effects model was that it was greater than 0, which means there was a positive association taking into account all these studies and their different weightages Over here, the glycemic load, we can also see that the diamond is against leaning towards the right, which means it again has a positive association, though very slight. And so from this test that we saw, there was Eggers test, which was used to test for publication bias. If you're unfamiliar with how statistics work, but basically when p-value is greater than 0.05, then our hypothesis is accepted. And in this case, it means that there is no publication bias. For Wall test, it tested to see whether the effect size was truly a positive and truly not one, which means that there was an effect of glycemic index, and we saw that the p-values were lesser than 0.05, which means the hypothesis was rejected, supporting the fact that there is a positive association. The heterogeneity test was also seen, and this was important since I've used the collective term of gynecological cancer, which means the studies may be weighting in terms of the methodology and the risk factors, the confounding variables. And so thankfully it was not very high. So there was moderate heterogeneity, which gives us the green light to truly combine them into a meta-analysis, making the results more reliable. So my overall conclusion is that there is a statistically significant association between dietary, GIGL, and gynecological cancers. And this is supported by the strengths of my study, which were basically that the study included a large number of sample size and number of studies, while also in the paper itself, there was multiple details on how the procedure was done, improving its replicability. And also, as we saw earlier, there was no publication bias. However, what were the weaknesses of the study? Well, in the study, we only considered the highest quantile and the lowest quantile of GI and GL. However, I did not produce a trend in how increasing GI and GL leads to increasing gynecological cancer risk. At the same time, there are many biases that are there in the studies that are collected, which sort of carry on forward to the meta-analysis that I conduct. And as we saw earlier, also the forest plot, the overall pooled effect size was really close to zero, which means that it is really sensitive to change and it is not foolproof. And also my conclusion does not fully line up with other studies. And there's a lot of debate on the subject as well. In the end, what is the significance of these results? Well, I believe that when cancer is seen as this sort of malicious entity with unknown causes arising from genetic mutations that are out of our control, there are indeed some variables like diet, which may easily reduce the risk of GCs. And this is especially important in improving the morale and especially empowering women at a higher risk of gynecological cancer. And so I believe through self-awareness campaigns and through doctors relaying this information through evidence-based medicine, there will be greater clarity on the extent to which the power that diet truly holds over diseases such as gynecological cancer. Thank you so much for listening.